Welcome back to UC Davis Live. I'm Satirius Johnson. UC Davis is home to a number of biological collections, from fossils and insects to pressed plants and yeasts. The campus recently celebrated Biodiversity Museum Day, when the public got to take a look at some of the collections. And joining us today are the curators of 3 of UC Davis's museums to talk about their collections and how they support research, teaching, and outreach on campus. Lynn Kimsey is a professor of entomology and director of the Bohart Museum of Entomology, which houses the 7th largest insect collection in North America with about 7 million specimens of arthropods. Ernesto Sandoval is manager of the UC Davis Botanical Conservatory. The conservatory's collection of live plants supports research and teaching on campus, as well as for other academic institutions, including K-12 schools. And Kyria Boundy-Mills is a microbiologist and curator of the Pfaff Yeast Culture Collection in the Department of Food Science and Technology. It's one of the largest public collections of wild yeasts in the world, including original yeast strains used in California wine fermentation. Thank you all for joining us today.
Thanks for having us.
Yeah, definitely.
Yeah, thanks for having us.
As a reminder for our viewers, if you're watching us live, we are taking your questions. Just leave them in the comment section and we'll work in as many as we can. So why don't we start off with a very basic question. What is biodiversity and why is it important? Ernesto?
Well, I grew up mowing lawns for my dad in LA. That was not biodiversity. We have an enormous collection of 4,000 to 5,000 species of plants. That's biodiversity. It's sort of the breadth of species across the world, or in particular regions, California, a region of Mediterranean climate biodiversity.
And why is it important to know more about it? And what kind of purpose does biodiversity serve, in our lives and in our environment?
Kyria, you want to touch on that one?
Why don't Lynn, you could jump in on that one.
All of these things that we're talking about provide ecosystem services. They provide us with food. They give us diseases. They interact with us every day. Without that, we would not be happy animals at all.
That's right.
And there's a lot of interconnectedness, and one species kind of depends on the other, and it's a system with a lot of moving parts that all need to kind of like work together.
Yeah, well, think about it. So you've got an example here. I work with insects. So bees are important pollinators of plants. But it's just not nectar and pollen they're collecting yeasts, which they have to take back to feed to their babies. At the same time, the plants can't function without the bees and therefore the yeasts. So it's all very interconnected. Yeah.
Right. Well, I think we're going to have a fun conversation today, learning about your collections. Lynn, let's start with you. And I know this might be hard to answer because there's just so much to choose from, but what would you say is a highlight or two of the museum's collection?
Wow, it is a little hard. Probably 2 things. We have an enormous mosquito collection. And this is valuable because oftentimes a species of mosquito might vector a pathogen, whereas other ones don't. So if you can't correctly identify them, it's kind of important. And you also need to know where they are. The other thing we have is really one of the world's largest bee collections. And again, like I mentioned with pollination services, bees are critical to the thriving of both the human sort of part of the environment as well as wildlands. So yeah, those are 2 important components, certainly, in the collection.
Right. And mosquitoes, I guess, they can be a vector for spreading diseases and things of that nature. So, learning more about them. And so, when it comes to mosquitoes, if I were to come to visit the museum and, want to look at your mosquito collection, what would I be looking at?
You'd actually be looking at a number of things because we have a large collection of the adults, probably, I don't know, maybe 50,000 specimens. We also have a very large collection of slide-mounted larvae and pupae. And so depending on what you wanted to see, we could pull out, all or some, or, it's a lot to look at anyway. Yeah.
And how does the Entomology Museum's collection support outreach and education?
Oh, well, we have sort of what I call a 4-way mission. And that is, obviously, we support the research community on campus as well as nationally and internationally by providing specimens housing vouchers for research programs, and so on. And you might also think of us as being a genetic lending library. But we also support teaching on campus. We also support, we do a lot of educational outreach programs. So we probably in a normal year would present programs to anywhere between 10 and 15,000 kids, parents, and teachers. And then we do information services. So if someone calls up and says, I have this murder hornet in my backyard, can you help me? And we look at it and go, yeah, that's a Jerusalem cricket. So, it's really 4 important components in what we do. And a lot of it's outreach.
It's really great. So you kind of also act, you act as kind of like a public reference library. Anybody, any member of the public can call you up or send you a picture of some insect that they're encountering, and you can help them figure out what it is they're dealing with. Absolutely.
We do this a lot, probably 3 or 4 times a day.
That's a really great service. And as far as the lending library goes, so if an elementary school or a high school wanted to do something in their curriculum, would they be able to borrow some samples from you to use in their school?
Yeah, so I have an outreach coordinator, I guess you'd call her, who we've put together a whole series of traveling exhibits that schools borrow. They go to county fairs, they go to other kinds of programs that people give. And it depends on the emphasis that someone wants. If they want California insects, we have that. If they want pollinators, we have that. Aquatic insects, things like this. So yeah, we do a lot of that. Our traveling get more mileage than I do.
And just a couple of weeks ago, we did the same sort of thing. Grad students were doing the equivalent of a science exhibit at a school, and they came and borrowed some plants. I sort of, gave them some tidbits of information on those plants, and then they had little 5-minute outreach sessions with a bunch of groups of kids.
That's really great. So yeah, let's talk about the Botanical Conservatory. Ernesto, what would you pick out as like a highlight or two?
Probably for visitors, our succulents, as well as we have a really large diversity of succulents that are recognized across the world. And then carnivorous plants, we have a really good collection of those. So really popular with the visitors.
I'm sure. So do you put on like little demonstrations with those, and do they kind of perform on cue?
That's what they do. Yeah. Venus flytraps, we have several that we cycled through, and those are always a hit. But also the sundews, which are one of the most diverse groups of species, the Droceras. They have the little sticky hairs. So I talked to them how there's the slimy sticky stomachs on a stick that catch the insects. And so, visuals to show people the importance of diversity and what they do across the world. Yeah. And
then there are also the infamous, I guess, the corpse flowers, right? Is that a carnivorous plant or not?
No, it's a flower that it actually. So many years ago, when I was studying biology, botany in particular plants, I realized that I need to understand insects because plants and insects are like this, right? Very connected. And so that flower makes a smell that attracts flies. Flies are major pollinators of plants, and so and also beetles are visit that plant as well. And so it sort of makes for an attractive, if you will, interaction of species.
Right. You should explain why it's called the corpse plant, right? Or the corpse flower.
Yeah, that's right. Yeah, so because the flower smells like something dead. Yeah. So and flies, can find that kind of smell 2 miles away. So it's a great way of luring in pollinators. Right.
And they bloom every so many years or something?
Every couple of years. We had one, Ted the Titan, that bloomed every 2 years for a while. It's been a little more erratic recently. But we just had one last year, and when it does bloom, it's only 2 days because it's got to bring the insects in, but it's got to get them to leave as well. So it's only a 2-day event, really.
Right. I've seen that those events tend to be very, very popular. A lot of people come to the conservatory to experience that blooming for I guess for whatever reason. It's just so rare and so strange, right?
Yeah, and and all our collections, we sort of have what I call those ambassador species that bring in and attract people to look at the greater diversity of organisms that we have, like the coarse flower or the carnivorous plants. Yeah, I mean, Curia has some yeast that are very important in our lives, right?
Yeah, absolutely. So Curia, why don't you tell us about a little bit about the yeast collection? I mean, it's not maybe as visually appealing as plants or insects that are easily accessible.
Well, it's a matter of opinion, but
so tell us about your love of yeast and why yeast is so important.
Yeah, well, before I get into that, I just want to put this into a broader context. These are just 3 of the many, many biological collections we have here at UC Davis. We don't know how many are here right now, but last time there was a major survey was in 1992. Somebody wrote a nice big catalog of the 100. Lynn was involved in writing this. There was 102 biological collections just here at UC Davis.
Amazing.
And because, so why do scientists collect and preserve these kind of specimens? And that's basically because of how science is done. We make a discovery and we have to be able to repeat that to prove that it was true. And then science is built by building on past discoveries. And one of the most important things that these biodiversity collections are involved in is documenting what species has been found where. And for example, where over history, where has a certain plant species been found? Which of the varieties of that plant are found in arid environments? And that was something that's been based on past discoveries and naming. These specimens are used for naming the species. Every biological publication starts with, here's the species we're working with. That species name is connected to a specimen in a biological collection. So that's one of the huge roles of these collections. So we've got a bunch of plants that we know can survive in arid environments. So now those can be used to develop crops that are tolerant, for example, for climate change. And then as we get more technologies, we can sequence the genomes and figure out what genes are responsible. So we go back to the collection and sequence the genomes. And then once we have genome editing tools like CRISPR, decades later, decades after these specimens were collected, we can go back and use CRISPR to make drought tolerant crops. So this all depends on having all these specimens for decades. And so some of the collections, I was just going through this catalog today, some of the collections we have here at UC Davis, we've got collections of Drosophila, we've got the Primate Center, which is important for human medical related research. We've got the National Germplasm Repository, which has 30 years ago, it had 4,300 accessions of grapes, stone fruits, walnuts, pistachios. Now, all these crops, we need to save living plants of them so that we can use them in the future. We don't know what challenges we're going to have in the future. So all of these,
and actually, the point that you make that these collections were started decades before some of this technology we have today. Like the people who started those collections probably couldn't even imagine the depth of information they would be able to glean from these collections.
Right. So, for example, you asked about, now getting back to the question you asked about yeast and why they're so exciting. So I manage this collection that has over 100-year history. So we have yeast that have been preserved since the 1890s. So yeast is a living organism. It's a single-celled fungus. And so they were collected primarily for this basic research, figuring out what species are found in what areas. So Ernesto mentioned the interaction of plants and insects, and yeasts are also very important in that interaction. They serve as an important nutrient source. They're high in protein. They're important for nutrients for the insects that visit the flowers. So we have yeast that were isolated in these discoveries of what role yeasts play in the plant and the insect life cycles. So they were collected mostly by Hermann Pfaff in past decades and other researchers in really basic research, figuring out what species was found where and how it interacts with other organisms in its environment. And now I'm using these same yeasts that were isolated for this really basic research. I'm using them in and other people all around the world are using them in all kinds of ways. One of my projects right now, I got some funding from the Good Food Institute, which is funding a project to find a fat substitute for plant-based meats. Right now we're using palm oil and coconut oil. And for the last 10 years, I've been studying yeasts that can turn sugar, they eat sugar and turn it into oil. And a lot of my past work has been funded for the biofuels industry. So these oil samples that solidify at room temperature, I kind of set those aside as duds because we don't want fuel for our car that solidifies at room temperature. But now I'm pulling those duds back out and looking at them to see if they might be a fat that could be in some plant-based meat substitutes. So these are yeasts that have been isolated 50, 30, 50, even 80 years ago. And now I'm using them in totally new and different ways that originally they would not, Hermann Pfaff would never have imagined that I'm using his yeast to make fat for a plant-based meat substitute. So that's how research goes.
Yeah, and I think when people think of yeast,
you don't know why they'll be useful in the future.
Yeah, exactly. And I think when people think of yeast, they think of bread typically. So explain what yeast is. It's bacteria, but how is it different than just bacteria?
So it's not a bacteria. So yeast is a fungus. So it's not an animal. Okay. It's not a plant. It's not a bacteria. It's a fungus. It's a single-celled fungus. There's a couple thousand species that are known. We have over 1,000 of those here in our collection. And they play important roles in nature. They are degraders. They don't have chloroplasts like plants do, so they have to eat other material, they eat dead stuff, basically. So they thrive in sugar-rich, moist environments like grape juice, like nectar of flowers, like the insect guts, where nutrients are constantly flowing through. So they're associated with other, specific types of plants or insects or other habitats. And because they can eat a lot of materials, a lot of dead plant materials, they can be grown in different ways. There's many different species of yeast, and some of them produce interesting chemicals. Some of them make nice enzymes, biofuels, pigments. And they can also be used as a host for making proteins from genes from other organisms can be put into yeast to make new products. So they can, in addition to the familiar uses, they can be used as starter cultures for beer, bread, wine, all these other fermented foods, biocontrol of plant pests, fungal plant pathogens. So there's 1,000 ways that these yeasts are being used.
It's very exciting. I mean, to think that, you can apply this to anything from maybe energy production to a sourdough bread or something. So I want to remind people watching that we are taking your questions. If you're watching us live, just leave them in the comments section and we'll work in as many as we can. Ernesto, the conservatory houses living plants, but it's not the only plant collection on campus. How does the conservatory complement the herbarium?
So we're a collection of live plants. The herbarium is a collection of dead pressed plants that are primarily used for research, but they also do support teaching. So in some of the teaching labs here on campus, the couple classes that are being taught this quarter, plant systematics and California floristics, they want to see a diversity of plants. And so we have live specimens that we help provide, but some specimens are so rare, you don't have live specimens of them. So you have an herbarium specimen for demonstration, but the herbarium specimens serve those functions that Kyria was talking about of historical documentation of, in this case, distributions of plants, primarily in California, but across the world as well.
Right. Interesting. Okay. And Kyria, I saw that your collection has a pretty robust website. Is there any place on campus where people can come to see the samples or is there any kind of exhibit on campus for people, like a physical place where people can come to experience the yeast collection?
It's not as visually appealing of specimens as some of the other collections on campus. If you wanted to see my collection, I would open a freezer and you'd see lots and lots of boxes of little tiny vials. So you know, the
Right.
physical collection itself is not too visually appealing, but we do participate in some of these outreach events, so you can learn more about the collection, especially Biodiversity Museum Day, which was a couple of weeks ago. That is one of our major outreach events where we interact with the public, and we bring in some specimens of yeast and petri plates, and people can learn about what yeasts are and how we're using them for our research, teaching, and outreach.
Right. Do you get a lot of requests from either restaurants or, food industry people when they're trying to come up with a new product or a new bread or anything like that? Or maybe they want to go back to an original strain of yeast, for their starter, anything like that? Any kind of, interesting stories?
Well, our samples are fairly expensive because we do sell specimens. Unlike the collections of dead specimens, when we have live specimens, we can grow them and distribute copies and keep our originals. So we do sell specimens. They're research grade, so they're like $150 for a little tube. So a home brewer, that would be really out of their budget. But we do work with, for example, a brewing starter culture company has licensed some of our yeasts. So we do work with companies that produce and market yeasts for a broader audience, for improving their strain catalogs.
That's great. So what are some things that would surprise people about your collections? Let's start with you, Ernesto.
That there are so many species in one space. We have a 3,000 square foot greenhouse, and it's only 14 feet tall and 30 by 100, so 3,000 square feet. But it's jam packed full of plants, and it really has an effect on visitors when they see all of that diversity in one place. They can be standing under a chocolate, be looking at a staghorn fern, at a bunch of orchids, including a vanilla orchid, understory forest plants, and they get a taste of standing in a rainforest somewhere.
Oh, that's great. Lynn, what about you?
I think people don't realize how many insects there actually are on Earth, and our collection probably only holds, it's a global collection, but we probably only have, I don't know, maybe a few 100,000 species, which is not much considering the current thought is that there are between 20 and 30 million different kinds of insects on Earth. And in California alone, there are about 100,000 species of insects. So wow. Our collection is very strong in the California fauna, but as you can imagine, most of those things are tiny, and people can only really sight identify the big things, butterflies, big beetles. The vast majority of insects are down around an 1/8 of an inch. You can't identify those in the field. So, I think people are surprised by that. And then they're also surprised by how many specimens of the same species we might have. So we may have 5 drawers of some kind of butterfly, for example, or a moth. That could be 4,000 or 5,000 specimens of the same thing. And people go, well, why do you kill so many? But the problem with insects is, except for these very charismatic big ones, we don't know where or when insects occur. So they're very seasonal as adults. They could be very patchy in distribution, and the distributions kind of shift over time. No population stays the same. And so the only way we can track this is actually to have a specimen with that information on it, right? So all the specimens have locality, collector, date, lat longs, if it's a more modern one, for example, so that we know where each insect occurs. And this has become even more relevant when we get new pest species coming into the state, for example. We can track the point of entry, how far they've dispersed over time, and this kind of thing. So it's definitely a working collection in a lot of ways.
And Kyria, what do you think, what are some things that would surprise people about the yeast collection?
Yeah, that's a really good question. It depends on the person. Some people are surprised to learn that yeast is alive, and that the yeast is a fungus. Yeah, you learned something new today. Yeah. So, but then other people in the field, one thing I'm constantly surprised in is how many different ways one yeast, I have 9,000 specimens, over 1,000 different species of yeast right now. And we're just starting a project to acquire another 5,000 specimens from a professor in Ontario that just retired a couple years ago. So this is a very large collection, but each specimen can be used in so many different ways. And I was just going through my database a while back, seeing what yeast I have used the most in just in my own research project. So we have one yeast strain that we've used for studying how much oil it can make when we feed it different sugars. It happens to be one of our pretty pink yeast. So we studied what are the carotenoid pigments, what's the chemical structure of these pigments. It's also one of our yeasts that we're very surprised to learn that it can secrete outside that cell. It can spit out this weird sticky goo that turns out to be a new type of detergent. It's a surfactant. So we discovered a new detergent. The same yeast, after you squeeze the oil out, what's left is high in protein. We could feed it to plants. There's companies that are interested in the surfactants. There's companies interested in how that surfactant gets squirted outside the cell. What's the secretion mechanism? Because if we can get other yeast to secrete their products rather than having to pop these little hard cells open to get it out of there, that secretion process could be really useful. So, those are just one strain that I've used in my lab for my project in so many different ways. And then there's another 9,000 yeast that we haven't even gotten into discussing about. So yeah, almost so I have 9,000 strains in my public catalog. Half of those strains I have sent out to some other lab in the world for them to use in research sometime in the last 20 years. So these yeasts are being used for really basic research, from discovering and naming new species, figuring out how yeasts interact with the plants and insects in their environment, to all these different applied areas, from food and beverage fermentations, food ingredients. There's so many different things that can be done with yeast.
That's so interesting. Before we go, I was just wondering if I could briefly ask each of you, what drew you to your field? Kyria, let's start with you.
Oh, what drew me to my field? So I started working here at UC Davis in 1998. I got my PhD studying one gene in one species of yeast, and I thought I knew a lot about yeast. And then I started digging into the culture collection world and realized how much I don't know about yeast. And so I had the honor of working with Herman Pfaff for 2 years. And I really, some of his, he was 88 years old, retired many years, and still coming to work every day because he loved yeast so much. And a little bit of that enthusiasm for yeast has rubbed off on me. And so that's what, working with Herman Pfaff is what really got me enthused about yeast, and having the responsibility of taking care of this amazing resource has really gotten me embedded here at UC Davis.
That's great. Ernesto?
Well, I think my father was a landscaper in LA, and he'd done farming back in Mexico and farming here in the US, farm work here in the US. And, I helped him out on the summers and weekends, and kind of got that kind of ingrained into me. And I was asking him questions driving along the roadside when I was a kid, what species, what is that? What crop is that? And so I had it there kind of, sitting in the back of my head. But like many other people in the world, I suffered from a certain amount of plant blindness, although I had, some training in it. And so I thought I wanted to study marine biology, marine organisms. I came to UC Davis to study biology in general, but I thought I would be a marine biologist or an educator, a teacher in a high school. Took a tour of the greenhouse, the conservatory here my freshman year, and just fell in love with the diversity and all the questions that could be answered about the way the world works with respects to plants. And I found my passion. I love looking at all different kind of plants and then helping the public understand how those plants work. And so I had some training in it, but then I came to UC Davis to study biology, got into botany, and then learned horticulture here at the conservatory, but melded my science background with my practical hands-on background, maintaining this amazing collection of plants.
That's great. And Lynn, what drew you to entomology?
I blame my parents. I got my first butterfly net at the age of 5, and started collecting butterflies like so many people who go into entomology start, and it just snowballed from there. I went on to college. Well, even in high school, I made collections, and then went on to college, ended up at Davis, and continued working on it. And I was always struck by the enormous diversity of what I was seeing and how little I understood it, and how impractical it was to really get a handle on what I was seeing. And so I eventually, under the tutelage of Dr. Bohart, or Professor Bohart here, focused on stinging wasps, and that's kind of where I've kept my studies ever since. And but I do a huge amount of, I guess I'd call it civil forensics, product contamination, things like this. It's so entertaining because you get to apply the research you do to something. It's a different kind of problem solving that we get than we have on campus. So years ago, I actually was involved in a homicide investigation because they wanted to know where the car had been based on the insect splats on the windshield and the radiator. That was awesome.
Wow. Yeah.
and it actually worked. But who knew? So it's the application of knowledge of the diversity of insects, their biochemistry, their behavior, their things, that is just, every year it's something new. I've been doing this a very long time, and it never gets dull.
Yeah.
And
well, it's exciting to know that, I was just going to say, it's exciting to know that there's still a lot of things to be discovered and to be learned.
Oh, we have, this is one of the problems with insects, a lot like what Curia has with the yeast, except you add a couple of zeros to it. I've already described over 250 new species of insects, and that's the tip of the iceberg. I mean, in California alone, if we have 100,000 species of insects, somewhere between 6 and 8% of them are new to science. And that's California. Can you imagine what it's like in the tropics? Just mind-boggling.
Right, exactly.
Yeah.
Ernesto, did you want to say something?
Yeah, when I was a student, I touched on this earlier, of plants, I realized that I need to understand insects because of the pollination biology and all the positive and negative effects that insects have on plants. And so a number of years ago, I'll say, is I took an entomology class, and I took it from Lynn. And I learned a lot about insects and how they work. And it helped me really put together more of the understanding of the importance of biodiversity and how the interconnectedness of species, in this case, particularly plants and insects. So, Lynn, you definitely bugged me in the right way.
I try.
Well, listen, this has been really interesting, really fun. I want to thank you all for coming on to the show today.
Thank you for having us.
Oh, yes, definitely.
Yeah, thanks for having us.
Mm-hmm.
Lynn Kimsey is a professor of entomology and director of the R.M. Bohart Museum of Entomology. Ernesto Sandoval is manager of the UC Davis Botanical Conservatory, and Curia Boundy Mills is curator of the Pfaff Yeast Culture Collection in the Department of Food Science and Technology. On the next UC Davis Live, how is Russia's war on Ukraine affecting supplies of energy and fertilizer? We'll hear from 2 UC Davis economists. That's coming up Thursday, April 14th at noon Pacific. I'm Satirius Johnson, and this is UC Davis Live.